Okay. Hello, everybody. It's Mrs. Shannon. This is part 3 of the first of the biochemistry Prezis. And the good news is part 3, carbon, is actually going to be the shortest one because I am going to cut out a bunch of information that you don't need to know in such detail as it is given here. So we're going to look at carbon here, and we're going to look at why it's such a big deal. So the big question here is, why is carbon a fundamental atom in biological systems, and how does the structure of carbon allow for different complex molecules? Remember, these complex molecules are emergent. These complex properties of the molecules are emergent. They have different properties than the atoms that come together. And so what makes carbon so cool, and that molecules that involve it are these complex, they have different properties, such crazy properties when compared to the carbon atom by itself and the other singletons that might be attached to it. And so biology is carbon-based. It is the primary element in our body. It is, in fact, it is so important, carbon is, that they have a whole line of chemistry, organic chemistry, devoted to it because it is found in all living organisms. It's very abundant and it's very versatile. What that means is that it can bond in a very stable fashion with many other elements. Carbon is tetravalent. It makes 4 bonds. It's happy when it's sharing its electrons and having covalent bonds with all these different other elements. It wants to make friends with all the other elements. And so these 4 bonds, as opposed to like 3 or 2, makes it have more possibilities in the combinations. I won't get into a big detail about why it's 4 and not 5, but hopefully remember that when looking at the periodic table, think about valence electrons. We have 8 in our outer orbital. If it has 6 electrons, for instance, in its outer orbital, it's very, very close to reaching its octet. It will try to pull 2 over, and you tend to make ionic bonds. Same thing if you're at the flip end. If you only have 2 valence electrons, you're more likely to give it up and drop down an octet. And so you tend to make ionic bonds. But if you're right in the middle there, if you have 4 valence electrons, you're happy with sharing. It's the easiest way for it to do it, to form a stable molecule. There is nothing special about life's building blocks, although it's still kind of cool how it could have come about. We'll be looking at a video next week about possibilities to the origin of life, and we're going to look at non-living systems to model a living system and how this might have come about. But there's this idea of vitalism, a life force that might have sort of started everything. It's a theory that there is nothing special about how life is built. Stanley Miller and Urey, they had a origin of life model that is well known here, experiment on how we might have had inorganic elements such as ammonia and nitrogen and water, and how they might have come together with an electrical spark to produce hydrocarbons and amino acids, these building blocks, these macromolecules needed for life. When Stanley Miller and his partner Urey were able to show this, the idea that there is a life force out there, that there is some higher power, kind of went out the door, at least as far as molecules go. Not saying that there isn't, but we saw their experiments let us see that there is a possibility of inorganic elements coming together to produce something organic. Now, this is the part I'm going to gloss through. They spend a lot of time in the book talking about isomers. This is the part you do not need to get bogged down in the vocabulary and what they are. Isomers are the same formula, different shape. So you could have 2 glucose molecules that have slightly different shapes depending on their structure. You could have 2 C2H6 molecules, ethane molecules that are slightly different in their structure. Any molecule more complex than propane has at least 1 isomer. It's another example of an emergent property. Actually, I said ethane could, and never mind. Ignore the ethane part. Any molecule more complex than propane can have an isomer. And so we can have, same formula, same amount of carbons, same amount of hydrogens, different structures, and they form different isomers. What's important about it is that because they are different structure, they will have a different function, even though it's the same elements building it together. It's an idea of an emergent property. Same elements, same properties coming together, but the way they come together are going to affect the new properties that they display. Biological systems tend to use only one of any of the two enantiomeric forms. Okay? And think about why this is. Think about structure and function. What would happen if it didn't behave this way? Here's the other thing. Functional groups. We're going to do this activity next week called pattern matching. You do not need to memorize functional groups. There's a list of functional groups in your textbook, and it's right here as well. Basically, a functional group is an identifying group. It's a characteristic of a molecule, and we're going to look at like amino acids and the functional groups in amino acids. It's a characteristic or characterized group, a group characterized by certain elements. So we have a hydroxyl group, which has OHs, which has an alcohol here. Okay, anything that has a little OH here would be considered an alcohol. You don't need to memorize these carbonyls, carboxyls. You don't need to know that. What you do need to do is start being able to recognize which functional groups are going to behave like hydrophilic molecules, which ones are going to behave like hydrophobic ones, which ones are polar, which ones are nonpolar, which ones are acids, they have a hydrogen that they can donate, which ones are bases, they want the hydroxide group, which ones are going to be ring-shaped. Oh, nah, not ring-shaped. Which ones are going to be aminos? They have a nitrogen in it. Which ones? We have the thiols with the sulfurs in it. We're going to look at cysteine, for instance. And so we need to be able to recognize when we have a sulfur there that it's going to behave a certain way. What we want to look for are the patterns within these. So you don't need to memorize the names of the functional groups, but start recognizing that there are differences in the way that they look. And when we start tagging them onto amino acids, it will affect the properties that the amino acids have, and in turn, the function of the proteins when those amino acids come together. So an example of this is these molecules are very, very similar molecules. There's just a couple of difference in the functional groups on these molecules, and that sets the testosterone and the estrogen apart. So small differences in the functional groups, big differences in the results. And it's all because of the way that they're going to interact with other molecules around those. And that's all due to the hydrophilic and hydrophobic interactions. So any questions? Key here is recognizing patterns, recognizing same and different functional groups, not the vocabulary. Don't get bogged down in that. Don't get bogged down in isomers, but understand that only one is usually found in a biological system. And if it is a different isomer, it is going to have a different function. Be able to explain how the structure of the carbon atom leads to the emergence, how it can have so many different varieties in combinations of elements that are bonded to it. And each combination, each structure could have a different function. It says identify all of the functional groups. You don't need to be able to say the names, but you need to be able to kind of recognize them in the future when we encounter them. And explain how each of the functional groups contributes to the properties. Again, it's because of that, the shape of the functional group, the polarity of it, and the elements that compose it. So if we have a polar molecule, a polar functional group, like an alcohol, it's going to behave differently than a non-polar functional group. Okay. So I hope this helps. Bring questions, send me questions, use your textbook, and we will go on to macromolecules next.